Good morning. Welcome to the second day of 3D SIG. I'm John Moult and I'm chair for the session. I sort of volunteered to be chair because I wanted to make some introductory remarks before we start on particularly the first three talks are really, for me, a very important occasion. So going back a bit, when Rafi and Ilan Shemish first asked me to help them start 3D SIG in 2004, I thought it was a great idea because at that point we had things like CASP and Capri, which were monitoring how well methods in this field worked, but we didn't really have a forum for exploring new ideas on a large scale. And to me, that was what this is all about. And I think it's worked very well. You come to this session, you hear lots of talks. Some of them you think, oh, I don't believe in that. And some of you think, well, that's interesting. Maybe I'll go away and work on it. And a lot of the things you hear about, in fact, at here, you never hear about again. They come, they go. That's fine, because it's an ideas forum. But the first three talks we're going to hear about are not just ideas. There's something which really works. I think all of you probably know something about this story already, that the application of fairly conventional convolutional neural networks to predicting three-dimensional structure has turned out to be very successful in the last CASP. You can look at the data for CASP. It's coming out now. You can already look at it on the website. And what you'll see is, first of all, if you look at contact prediction, the accuracy with which contacts be predicted, which for many years in the particular metric we use was stuck at 20% precision, so 80% false positives, 2 CASPs ago took a big leap to about 45% precision because of the introduction of more sophisticated but conventional statistical methods. What happened in CASP 13 is it went from 45% precision to 70% precision. So they were down to 30% false positives. You can really start to do stuff. And that change came from the application of conventional sorts of deep neural network. And of course, as many of you know, it's not just predicting contacts. The networks are able to go beyond that and predict distance distributions between pairs of residues. And so it's as if you have a potential of mean force between every pair of residues in a structure. And for the groups who use that effectively, that really led to, I would say, it's a big statement, but I think it's true, the solution of the classical problem of trying to predict the fold of a protein when you don't have a template. So the classical folding problem for monomeric proteins, and there are caveats, is essentially solved by these methods. If you look at the CASP targets that didn't succeed, they usually have some kind of intimate interaction with subunits and so on, and in many senses that's the next challenge in where we're going with modeling. But the sort of classical problem, the thing that got me into CASP 25 years ago, is solved to a large degree by these methods. They're not perfect structures, but they're correct folds. So this is a really, really big deal, I think. And so it's great that we're going to have 3 talks on this. Many of you, of course, will be aware of the publicity that's surrounded this and have heard about DeepMind's contribution. And there's no doubt that DeepMind did deliver, on average, the most accurate structures. And Andrew Senior is here today. I'm sure he'd be happy to talk to people about their work. I think it's important to remember as well that the ideas that led to that really developed over a number of rounds of CASP, from my perspective, amongst the CASP community. How can you take the ideas about deep learning and apply them to the contact and distance prediction problem, and then how can you go from that to structure? So the 3 talks we've got are going to be very much on that, and we're going to start with Jinbo Xu, who has really been one of the leaders in this. I have to say, Jinbo, over the years, you know, you had a lot of ideas, and I've said, yeah, well, that may work, I don't know, where is it? And then finally, it really does work. And so I think it's going to be very interesting to hear what you've got to say, and please take over.
Okay, thank you very much. And I'm very happy to come here to talk about how we can apply deep convolution neural network to protein folding. So in this talk, I will mostly focus on the template-free modeling. So before we know, the fragment assembly is a very popular method for template-free modeling. And this method works on some small proteins, but you have to do extensive conformation sampling, even for a very small protein. So in recent years, there are some there's a new method called contact assistant folding, or especially in the last 2 years, now we come up with the distance-based folding method. So here, this picture shows you the procedure for the contact assistant or distance-based folding method. So here, first we will do homology search. We find the sequence homologs for the target, and then build a multiple sequence alignment. From this multiple sequence alignment, we will do convolution analysis, and also we'll build the sequence profile. So we have both sequence profile and also the convolution information. Then we will feed them into the convolution neural network to predict both the local structure and also, more importantly, the contact or distance between the two amino acids. So based on the local information and also the pairwise information, then we can reconstruct the 3D modeling. So basically, this method avoids extensive conformation sampling and can deal with very large proteins and can also fold the protein much faster than the fragment assembly. So for contact prediction, it has a long history. So many methods have been developed for contact prediction. So we can group all these methods into 4 categories. So it's easy to see the difference between the co-evolution analysis method and also the supervised machine learning method. So the co-evolution method basically will focus on one multiple sequence alignment and then do co-evolution analysis to identify the co-evolved residues. Then usually the co-evolved residues, they have a good chance to be in contact. And the supervised learning method, they will take some features as input, like the sequence profile and also convolution information as the input. And then we'll do a supervised learning to predict if two residues will form a contact or not. So on another dimension, we can also divide all this method into either local methods or global methods. So for example, for the convolution analysis, the mutual information is a local method. The reason why we say mutual information is a local method because when we studied 2 residues, and the mutual information will only use information in the 2 columns in the multiple sequence alignment and ignore the coupling of all the other residues. So this is why we call this method the local method. And the global method we are making use for all the information. So we consider all the possible couplings in the whole contact matrix to predict if one pair of residues form a contact or not. So this is why we call this kind of method as a global method. So there are many global statistical methods developed for co-evolution analysis in the past 10 years. So similarly, for the supervised learning method, we can also divide all these methods into local method or global method. So before the rubber X contact comes out, basically almost all the methods are local methods because when they do the prediction, they only use information in the 2 columns and also ignore the interaction of these 2 columns with other columns. So all these methods are local methods. And so rubber X contact is the first effective global method for the supervised learning, but now there are also many similar tools. So all the contact prediction methods, all the most powerful structure prediction methods starts from the multiple sequence alignment. So we can build such a multiple sequence alignment by running some homology search, like we can use SciBlast or HHBlitz and also jackhammer. Then we can do homology search from some sequence databases, such as a non-redundant sequence database or UniCluster or UniProt. So from the multiple sequence alignment, we can do convolution analysis. So this is just based on a very simple observation. So if 2 residues they are close in the space, then if one residue mutates, then the other one is also likely to co-mutate. So this kind of convolution will reflect in the multiple sequence alignment. So when you look at the co-evolved residues, then you can, when you look at the two columns corresponding to the co-evolved residues, you will see usually these 2 columns they are highly correlated. So based on this observation, then we can do the co-evolution analysis. However, although in the past 10 years, the convolution analysis significantly improved, but this method still has some limit. So this method, in order to work, this method needs a large number of sequence homologs in order to work. Or even if you have a large number of sequence homologs, you can only recover a small percentage of native contacts. But when we look at the PDB and also the PFAM families without solved structures, then we will see a very big difference between the PDB and the PFAM families. So this picture shows you the statistics of the family size of the proteins in the PDB. So you can see the median size is about 680, which means for proteins with solved structures, actually we have quite a lot of sequence homologs. But when we look at the PFAM families without solved structures, the median size of family is only about 100. So which means for many of the PFAM families without solved structures, actually we don't have so many sequence homologs. So this means the pure convolution analysis method doesn't work very well for those PFAM families without solved structures. So we need a new method to deal with this kind of families. So different from the convolution analysis, the supervised learning method will take some input features and then do a supervised learning to predict contacts. So here is a typical procedure used by previous supervised learning method. So suppose we wanted to predict contacts between I and J, then we will take some sequence features for position I and for the position J. And sometimes some methods also take the pairwise features for this pair of residues. Then we'll input all these features into the machine learning model, like a neural network, or separate vector machines, random forests, or even deep belief networks. Then this machine learning models will predict if there's a contact or not between these two positions. So this is a typical procedure used by previous supervised learning method. But as I mentioned before, this kind of method is a local method because it ignores the impact of all the other residues. It only focuses on two residues, only focuses on the coupling of two residues, and ignores the coupling of these two residues with all the other residues. So this is the issue with this method. So to deal with this issue, so I came up with some new ideas later in 2015. So I wanted to model the whole protein, or even at least a big chunk of the protein in a single system. By this way, so I can predict the neighbors or predict the contacts of all the residue pairs simultaneously. So this is very important. And then I wanted to use contact patterns and even global conformation contacts to predict the neighbor of one residue pair. So based on these ideas, then I formulated this problem, the contact prediction problem as an image semantic segmentation. So we can treat the contact matrix of the protein as an image, and each pair of residues as a pixel. So basically, we wanted to assign or predict a label for each pixel in this image. So for each pair of residues in this contact matrix. So this problem in the computer vision community is called the image semantic segmentation. So there are many deep learning algorithms developed for the image semantic segmentation. So we can borrow some ideas from this community for protein folding. So finally, I will employ the convolution residue neural network to the whole matrix. And I can build a very deep networks to capture very long-range contacts. So this is a new ideas for the contact distance prediction. So I started to develop this idea later in 2015, and then started to do the coding early in 2016. And I didn't finish the coding before the CASP 12 started. So I finished it at the end of the CASP 12. And so after this, after CASP 12, and I put up one paper on the bioarchive. So this is the paper I have to describe this kind of deep learning method. So this paper was officially published in January 2017. So this paper first showed the contact prediction can be significantly improved by deep learning. And also, in this paper, we show by using the predicted contacts, we can fold some very hard, also large proteins in the CAMIA experiment. CAMIA is the online benchmark, which is very similar to CASP. And then in 2017, I published another paper to demonstrate, actually, this method also works very well for membrane proteins, even if we don't have lots of training data for membrane proteins. And in 2018, I further showed this method also works very well for complex contact prediction. The nice thing is you don't need to retrain your model. You only train your model using the single chain and the serial proteins. And this model works very well for memory proteins and also for the complex contact prediction. So this is a nice thing with this deep learning method. So this paper has been downloaded by about 10,000 times, and early in 2018, so it received the Plus CB Research Prize in the category of breakthrough and innovation. So this is the first paper I have for using the deep learning to do contact prediction. So the method is called the fully deep convolution residue neural network. And there are some keywords like the convolution and the residue in this method. So here I briefly explain what are these keywords. So what's convolution? So convolution is used to detect patterns and also to collect information. So suppose convolution actually is very simple. It's just a matrix operation. So here, suppose we have input matrix, and then we have a kernel, also called a filter here. So basically, we scan through this input matrix using the kernel matrix. And at each position, we will look if there's a pattern in this position or not. So this filter basically is used to capture patterns from the input matrix. So when you apply more and more convolution operation to this input matrix, then we will collect information in the whole matrix. So this is the convolution operation. Then the next keyword is residual. So this is a trick to train a very deep learning model, very deep network. So in order to predict y from x, so we first predict y minus x from x. Then we add x and then predict y minus x to get y. So here y minus x is a residual, which means we don't directly predict y, but instead we predict the difference between y and x. So this is equivalent to add a shortcut between the input and the output. So this makes the training of the deep network much easier. So by using this residual module, then we can easily stack many convolution layers, like say 100 layers, to form a very deep neural network. Now I'm going to tell you some details of my algorithm. So here is the input. So the input is a multiple sequence alignment. Then from the multiple sequence alignment, we will derive two kinds of features. One is the sequential features, like the sequence profile or the predicted local second structure information. And then we also derive the pairwise features, like say we can compute the mutual information between two columns, and we can also run the direct coupling analysis to compute the direct information between two columns. So we will fit these two kinds of information into our deep network, and finally the deep network will predict the whole contact matrix or the whole distance matrix. So here is the details of my deep network architecture. So we have two inputs here. This is sequential feature and this is pairwise features. So starting from the sequential feature, we will apply the one-dimensional convolution or the one-dimensional residual network to the sequential feature and get the convoluted sequential feature. Then we add this, we convert this sequential feature into pairwise feature. Then we add this into the original pairwise feature to form a new pairwise feature. Then we will input this new pairwise feature in the two-dimensional dilated residual network. And then we will finally get the predict contact or distance matrix. So in this architecture, so this two-dimensional convolution is the most important component. So we use many convolution. Like say, initially we use about 60 convolution layers here. And the one-dimensional helps, but is not the most important thing. And now some groups, they even ignore this one. They just directly convert the sequential feature into a pairwise feature and add them into together to the original pairwise feature. So you don't have to do the one-dimensional convolution here for the sequential feature. So this is the network architecture we have for the contact or distance prediction. So we test this method in CASP 12. So as I mentioned, so I didn't finish it. I only finished the coding at the end of CASP 12, but still, this method managed to be ranked first in the CASP 12, although it's not much better than the metacycle, a very successful traditional neural network method. And in CASP 13, now quite a few groups, they implement this algorithm, this deep residual network algorithm for contact prediction. And so in CASP 13, now we did much better than the traditional neural network method. And so here, this is the top 3 groups for the contact prediction in CASP 13. Actually, so this picture shows you that Z-score just artificially amplifies the difference between groups. Actually, these top 3 groups, they are not very different in terms of the F1 value. So this picture shows you the progress we made in the past 8 years. So this CASP 10 result is about 20% of precision. The CASP 11 is about 27, something like this. And the CASP 12, we have 45%. So from CASP 11 to CASP 12, I think the improvement, half of the improvement comes from the algorithm improvement, from the algorithm, and half I think is from the database enlargement. And from CASP 12 to CASP 13, so we have another big jump of the performance. Now, this time, the improvement mostly comes from the algorithm development. So this is the progress we have in the past 8 years for the contact prediction. So which means now we can do much better contact prediction than before. Then a natural question to ask is, so some people will ask, if deep learning can really predict novel contacts or not? So we have to answer this question. So some people will suspect deep learning can only just simply fill the contacts in a second structure, so not produce a novel contact. So here I show you some examples, show you 2 examples to show you actually deep learning sometimes can predict novel contacts. Like here for the CASP 13 targets here. So in this picture, so in these 2 pictures, I use the gray point to represent the native contact. And the correct one is in red, and the incorrect one is in green. So in each matrix, this part shows you the prediction made by a pure convolution analysis method, the CCM, so which is one of the best convolution method. And this lower part is my prediction. So you can see here, for this target, the prediction made by CCM just contains too many false positives. The green point just spread over the whole contact matrix. But in my prediction, there are very few false positives. So this is one observation. The other one is more important here. Look at here. In this region, so CCM even couldn't predict any correct ones. But look at here. So in this region, so RapidX can predict many true contacts, can recover many native contacts. So this means by using deep learning, we can indeed identify some novel contacts which the convolution method couldn't identify. So this is one example here. And similarly, this is another example. By the way, for this target actually doesn't have many sequence homologs. It has about just a little more than 100 sequence homologs. And it's a very large protein, has more than 300 residues. And for this one, this example, you can see here, like in these regions, the CCM just failed to identify any native contacts. But the deep learning can identify many true contacts. So these 2 examples show you, actually, indeed, we can, by using deep learning, we can predict novel contacts instead of just filling contacts in second structure. And I also did some statistics. So I look at the entropy. So entropy is used to measure the spread out of the predict contacts. So I look at only very few. Look at just top 10 predict contacts in order to compare with the convolution method fairly. So I download the entropy score from the CASP 13 website and then compare RapidX with just one convolution method here. So you can see, even if we only consider a small number of top contacts, still the contacts predicted by RapidX has a better entropy score than the convolution analysis. So this means indeed the deep learning provides some actual information than the convolution analysis. So I just show you the results of contact prediction. Then we can easily extend the algorithm to distance prediction. Conceptually, the contact is only binary representation of distance. So if a neural network can predict contact, of course, it can also predict the distance. So not much work has been dedicated to distance prediction, but my group indeed started to work on this prediction in 2012. So in 2012, we developed a traditional neural network method to predict distance distribution and then derive a distance-based potential from this distance distribution. And we applied it to study degree ranking. And then in 2016, one of my former PhD, he studied the folding simulation using this kind of distance-based potential. And in the 2017, my +CB paper, I proposed to using the residual net to predict distance prediction as a future work. Then in 2018, we work on this. So we published one threading paper. In this paper, we describe how to use the residual net to predict distance and convert to a distance potential. Then we demonstrated this kind of distance potential can significantly improve protein threading. Then at the same time, also work on the folding using the this kind of distance prediction to fold a protein. Then in CASP13, we tried several different strategies. So we discrete, for example, we discretize. So we discretize the distance into 25 bins, except the first and last one. Each bin has 0.5 angstrom. Then we also tried 12 and 52 bins. So in addition to the C-beta distance, we also predict distance between like C-alpha, and nitrogen, oxygen, and also atoms. So the C-beta distance is most useful, but the other distance information will also help a little bit, not too much. So this is how to extend the network to do the distance prediction. So here I show you two examples. So like this one, this target has many sequence homologs, has more than 1,000 sequence homologs. So in this picture, so this upper part is the predicted distance information, and the lower part is the native distance. Here I only show you the predicted distance less than 15 angstrom. So you can see the predicted distance matches very well with the native distance. So for some targets, we can do really good distance prediction. Here is another example, and this target doesn't have many sequence homologs. But again, you can see the predicted distance information matches very well with the native distance information. So we know, so the sequence homologs, information in sequence homologs is very important. So here, I just want to show you the dependency of the contact prediction and the distance prediction on the depth of the multiple sequence alignment. So the first picture shows the dependency of contact prediction on the multiple sequence alignment depth. So you can see there is some dependency, but the dependency is not very strong. So the correlation is not very strong. So this is for contact prediction. And for the distance prediction, the dependency is even weaker. So the trend line R square is a very small value. So this is the dependency between the distance of contact prediction on the multiple sequence alignment depths. So this picture shows you the relationship between the 3D modeling quality and the contact and distance prediction quality. So here I show you the red point represents the contact precision for each target. And this blue points represent the distance F1 value and also the model quality. So you can see there's a better correlation between the model quality and the distance F1 value here. So which means if you can predict the distance very well, then you should be able to build the 3D model very well. So this is dependency model quality versus the distance quality. So here I show you the progress from CASP12 to CASP13. So since quite a few groups adopted the deep residual network method for contact or distance prediction, so in CASP13, we achieved, the whole community achieved the widespread progress. And so I think it's the largest progress in the history of CASP. So here I show you the progress in the server group and also in the human group. So here I just listed the top two groups here. So from CASP12 to CASP13, so you can see these two top groups on the free modeling targets. And then in CASP13, we have these top two servers in the free modeling category. So you can see from this one to this one, the improvement is about 0.1.5 in terms of TM score. So TM score ranges from 0 to 1. And compared to this one and the X, the improvement is about 0.1.3. So the improvement is very significant even for the server group. And this is the top two human groups in CASP12 and CASP13. So the AlphaFold improved by about 0.19. Then the human improved by about 0.1.3, which means AlphaFold speed up the progress by about 50% in terms of the TM score. So this is the progress from CASP 12 to CASP 13 in terms of the 3D modeling accuracy. So yeah, since as I mentioned, so quite a few groups has adopted the deep residual network method for contact distance prediction. Then what's the difference for the 3D modeling? Right, so here I pick up the 4 representative groups. So hopefully I can show you the difference among these 4 groups. So first one is Baker. So it's a fragment assembly method, but Baker also uses, for some targets, also use context predicted by the convolution analysis. Then Baker will build the model by using the sampling strategy and follow up by the gradient-based optimization, energy minimization. And some group predict context by using the deep residual network, and then do confirmation sampling, also fragment sampling to minimize the energy, to do energy minimization. And the AlphaFold predict distance by using residual net. And AlphaFold will also do both sampling and also gradient-based method to do the energy minimization. And then my group, RapidX, do distance prediction using the deep residual network. But instead of do the energy minimization, I simply use the distance geometry to build the 3D model. So I use a tool called CNS. So CNS didn't do the real energy minimization, just using the distance information to build the 3D model. So this is so you can see the difference. Like so compared to Zhang and Baker, so AlphaBet is better since AlphaFold did distance prediction. So AlphaFold has more information from the deep learning step. And compared to RapidX, so AlphaFold is better in building the 3D model. So this is why AlphaFold stands out in CASP 13 when doing the 3D modeling. And this table shows you the contact prediction accuracy in CASP 13 of these 3 groups. So you can see in terms of the contact prediction, actually the difference between these 3 groups are not very big. Yeah. So these 2 groups are slightly better than Zhang. Yeah. So in terms of contact prediction, which means in terms of deep learning, in terms of deep network architecture, the difference is not so big. And here, just to show you the latest results by Baker Group. So it's this result is developed. This program is developed by Professor Yang, who is now a visiting scholar with the Baker Group. So they also implemented a deep learning algorithm, use deep residual net to do the distance prediction, then feed the distance into Rosetta. So this is the folding results on the 32 CASP 13 free modeling targets. So you can see Rosetta now can also do the 3D modeling very well, as long as you have the distance prediction by using the deep learning method. So this is the latest development in this field. So now I'm going to show you some specific examples. So here, at the beginning I mentioned by using this kind of deep learning method, now we can fold very large proteins. So here I'm showing the results on 3 largest targets in the CASP 13. So these are the 3 targets I wanted to show you. And these 3 targets has about 350 residues with valid coordinates, so much larger than the first one. So here is the sequence length, and here is the depth of the multiple sequence alignment. Then here are the TM score of the models by 3 groups. So you can see here, this target is the most difficult one. It's a large protein, but it only has a little more than 100 sequence homologs. So it's a very challenging target. So although our folding method is quite simple, we just use distance geometry, but actually we built a pretty good model for these 2 targets. So for these 2, we did very well in terms of 3D modeling. And for this one, we did very badly compared to the other 2 groups. So for this one, very hard, this target, so actually only 7 models submitted for this target has TM score larger than 0.5. So 5 are from RapidX, 1 from Zhang group, and 1 from Chen Smarticon. But this model actually is just a copy of 1 for RapidX model. So this is the model for this very challenging target. But now by using energy minimization, I can boost the TM score from 0.58 to about 0.7. So by using energy minimization, we can significantly improve. So I can significantly improve the modeling for this target and also this target. But for this one, still, I couldn't beat my distance geometry method. So energy minimization is not slightly worse than the distance geometry method for this target. So here I show you some pictures of these 3 targets. So this is the modeling of this target. So you can see basically at the fault level is correct, although the resolution is not very high. Yeah. So this is for another 2 targets. So in CASP, I'm not aware if there's any membrane proteins which are hard. So in CASP 13, we have some membrane proteins, but it looks like they have templates. So I just go to Cameo. So I take some examples from Cameo. So here I show you 2 examples. These are 2 hard targets in Cameo, and except our server, all the other servers failed on these 2 targets. So this membrane protein with about 300 residues. So we get a very good model for this target. And this target doesn't have similar folds in PDB. So this is another example. So this confirms, so the deep learning algorithm also works very well on membrane proteins. So you don't have to train the model using membrane protein. You just train using proteins in PDB, then it works very well. So I just show you now we can do 3D modeling on hard targets very well. But yeah, you may ask, so why the deep learning work? So can deep learning really predict the novel fold or just a better fold regulation method? But actually, even if for CASP hard targets, some of them have similar structures in PDB, although at the sequence level, they have no relationship. But you can indeed find similar structures for some hard targets in PDB. So it's not very easy to answer this question, to tell you if the deep learning can really predict a new fold or just recognize better fold from the PDB. So I did some study using my own data here. So first, I measured the structure similarity between the CASP 13 targets, free modeling targets, and my training proteins. I measured the structure similarity. Then I also looked at the quality of the model produced by my program. So this picture shows you the correlation. So this curve, this line shows you the correlation between the model quality and the target training structure similarity. So you can see, first, the correlation is very weak. Second, the correlation is not positive, it's negative. So which means when the target is more similar to the training protein, but actually the modeling accuracy is not so good. Yeah, the modeling accuracy doesn't increase along with the similarity between the target and the training proteins. So this is the correlation between model quality and the target training structure similarity. And this red line, so all the points, all the targets above this red line indicate the model quality actually has a higher TM score than the target training structure similarity. So hopefully this picture can convince you actually for some targets, the deep learning indeed can predict new fold. So deep learning not simply copy from the training proteins. Yeah, so this is one picture show you deep learning indeed can predict new fold. And then I did a further study. So I measured the structure similarity between the models produced by LaPleX and also the training proteins. So look at how similar are the models with the training proteins. So this is the distribution of the TM score. So you can see for most CASP 13 free modeling targets, the similarity between my model and the training protein actually is quite small. It's less than 0.55, which means for most targets, they are not similar to the training proteins, even if you do the structure alignment. This also means the model built by deep learning by my algorithm, indeed, they are new fold. So they are not simply copied from the training proteins. Yeah, so hopefully these two slides can convince you actually deep learning indeed can produce a new fold instead of just copy from the training proteins. So we know the direct coupling analysis is important for convolution analysis. It significantly improves our capability to detect co-evolved residues. But for CASP targets, how important is the direct coupling analysis? So I did some study on CASP 12 hard targets. So first, I test on the contact prediction. So I trained one model using both mutual information and also the information produced by direct coupling analysis. Then training my model uses the mutual information only. I don't use the information produced by the direct coupling analysis. So indeed, the contact prediction accuracy drops, but not too much. It's by about, on average, about 7%. So which means for cusp targets, it's fine, even if you don't do the direct coupling analysis. So I think the major reason is because many cusp targets, they don't have a large number of sequence homologs. So which means in this case, the direct coupling analysis is not very useful. Similarly, for folding, so you can see also, yeah, the model accuracy drops a little bit by about 0.6 if you don't use the information produced by direct coupling analysis. But the model result is okay, it's reasonable, even if you only use the mutual information. And so in my impression, David John's group and the Young group, they also did some similar studies. So they used the covariance matrix as input, and they can predict the context very well, even without using information from the direct coupling analysis. So this is the importance of the direct coupling analysis for cusp targets. I think for the proteins with more sequence homologs than direct coupling analysis may be more useful. So in cusp sorting, John Mould asked one question. So why deep learning didn't perform much better before? Why we have to wait until cusp sorting? Well, so here I try to answer this question. So actually, a simple answer is not all deep learning methods they are equally important. They are equally powerful. So in 2012, so Chen group developed deep belief network for contact prediction. In cusp 10, this method better than the random forest by about 1.2%. And in cusp 11, cusp 12, is even underperformed the traditional neural network method meta-side curve. So which means when you use deep learning, for this problem, the DBM method is not very effective. So, and then only until 2016, then once we have the fully convolution neural network, then we can significantly improve the contact prediction, and also we can do folding much better. So by the way, so CNN and DPN, they are two different deep learning techniques, so they have no relationship. And actually, CNN was invented much earlier than DBN, so which means sometimes actually old techniques may be more useful than new techniques. Okay, so I will summarize my talk. So yeah, now we have significantly improved time-free modeling. So first, we have to thank. We need to thank those people who have produced the large amount of data. So we should thank those people who produce large amount of sequence and also the structured data. Without this data, we couldn't train our deep learning model. And so the key ideas underlying this progress are two key ideas. The first one is convolution analysis, and the second one is deep residual. But both methods have a common property. So they will model the whole proteins, or at least a big chunk, in a single system. So you shouldn't look at just two residuals independent of the other residuals. It's better to model the whole proteins in a single system. So this way works best. So I think this is more fundamental than you use the deep learning. In the future, maybe we will have better techniques, but I think in the future, we still have to model the whole protein in a single system. Yeah, this is more important than the deep learning technique. So by using deep learning, we can fold both small and large proteins, and then we can fold much faster. So like say, within 4 hours, I can fold all the 37 12 fully modeling tasks on a single Linux machine with 20 CPUs. So we can really do folding much faster than before. And finally, I just want to point out two references. So some of the results are in just two papers. So they are not in my, so these two papers are in bioRxiv, but they don't contain the results I showed in just slides. So this paper will be published very soon. So the conference version is out, but the general version will be published very soon. And this one is still under review. Okay, I will stop here. Thank you so much.
Okay, questions? Please use the mics. There are 4 mics. This one's not here, but David.
Jinbo, a very nice talk and some nice results. I guess I'm just going to raise one issue, because I think if we're going to try and improve these methods, we kind of need to understand kind of what's going on. Your definition of local versus global methods, I think in the most part, I think is wrong, because most of the things you label as local methods actually use global methods as their input features. So they're already using all of the coupling information from all the residues as their inputs. And I don't believe it's possible to apply a neural network to those inputs and convert it from global back to local again. So I understand what you mean about the filtering being larger scale, but I think inherently the inputs to those local features, into those methods, most of them, I think, at least, so Psychov going to MetaPsychov, for example. Psychov's in MetaPsychov, so MetaPsychov can't become local once you've already labeled Psychov as being a global method. But I mean, I guess, and I hate making comments without questions, so I was saying, please comment and please disagree with me if you like.
Yeah, just a very good, very great comment. So first, I didn't say Psychov is a local method. Psychov is a global method. MetaPsychov itself is a local method. The input is global, but the network architecture is local. This is what I meant. So I don't mean the input. But as I already, you already have one paper, and also show you, even if the input is local information, but using the deep network, you can get a very good contact prediction, right? So using the covariance matrix or use speech information as input, actually we can do contact prediction very well. So even the input is local, but using the deep residual network, but using the global method, we can do contact prediction very well.
Let's go alternate mic.
Hi, very interesting talk, thanks. When you say large proteins, what do you think is the current size limit for applying these methods?
I guess the limit is, so it's hard to have a very exact definition for the limit. So it depends on the trade-off between the size and also the number of sequence homologs we have. For large proteins, usually we will need more sequence homologs in order to be able to work. Yeah, so we should look at the ratio between the multiple sequence element depth and also the sequence length.
Yeah, exactly. You got the slide I wanted to ask about. Yeah, so that's you claim that mainly that the CNNs is much better than DBNs. But for me, the big difference is that you, in your 2016 implementation, you used DCA as an input, and you did not use that 2012. So have you tried to take out DCA from your CNN, and have you tried to add DCA to the DBN?
So I didn't try DBN never. I never tried DBN, but I tried another strategy. So instead of apply the residual to the whole matrix, I just take, so just similar to the previous supervised learning method, take a small matrix, and I use the residual to predict the neighbor of this matrix, just predict one neighbor, just formulate as an image recognition problem. And this method actually doesn't work very well. It can improve over previous method, but the improvement is not very big.
Oh, hi, a very nice talk. Thank you so much. I have a small question. So how do you enforce like global constraints in your like say contact or distance predictions? For example, if amino acid is in alpha helices, it's unlikely to be in a parallel beta sheets or in parallel beta sheets. And you have very clear patterns in the contact map matrix. How do you enforce that kind of global constraints?
This is a very good question. Actually, I'm also thinking about this problem. So right now, the deep learning method will implicitly learn the global constraints. But we only explicitly enforce the constraints when we build the 3D model. Yeah, but I think it would be better if in the deep learning step, we can explicitly enforce the global constraints into the whole matrix.
So you think that the results that there's good patterns is actually a coincidence, not by design, that your deep learning actually can do that? It's
yeah,
not.
deep learning can implicitly learn some constraints. Yeah.
Right. Yeah. Thank you.
Yeah.
Hi, thanks. I have a similar question to Arne's one. So you are not using DCA.
So I use DCA. DCA is useful. I didn't say DCA is not useful, but I just want to tell you how useful is DCA for the CASP targets. Yeah.
And can you comment if other contact predictions methods are using DCA prior to applying the convolutional neural nets?
What kind of neural network? You mean using DCA plus convolution, right?
Yes.
But actually, I used both DCA and also convolution. Yeah. Yeah, I would say most methods use DCA information. So DCA is useful. I didn't say it's not useful, but I just want to try to quantify how useful is DCA for CASP targets.
Okay,
yeah.
Thanks.
Two more questions.
Hi, thank you for the talk. So you've given some analysis of convolutional neural net performance with evaluated against deep belief network. So there were some submissions to CASP13 that used recurrent neural networks. Have you ever looked at recurrent neural networks, how they work or don't work, or why? Can you comment on that?
Actually, I tried. I myself tried a recurrent neural network. So I add a recurrent neural network onto the convolution network. So improvement is not very big. So in terms of contact prediction, it's 1 to 2%. And there also report from another group, from the group. He reported about a 5% improvement by adding the recurrent neural network onto the convolution network.
Hi. Very nice work, clearly. Everyone can understand the logic behind contact prediction, right? If, as you mentioned now, if you change from a small amino acid to a large and you see the connectivity, the other one should change from a large to small, etc. What is the logic? I know that it's quite a philosophical question. What is the logic that this kind of information will influence the question, what is the distance difference between a bin of 10 angstrom to 10.5 angstrom? What in the structure make this kind of relationship relevant?
This is a very hard question. Yeah, it's really hard. Actually, yeah. So we know it's hard to understand deep learning. But what I can tell you is, so in addition to the co-evolution information, our deep learning model also indeed learned some structure information from training proteins. So deep learning will learn some structure patterns from the training proteins. Then by combining both the co-evolution information and the structure patterns, then we can, for some proteins, we can roughly determine the distance of some proteins.
All right. Thank you very much again, Jinbo. Sorry to cut off the discussion, but we have to stay on time.